Okay.
Gülsen Meral Hocam, siz epigenetik konusunda Türkiye'deki uzmanlardansınız. Başta gelen uzmanlardansınız. Epigenetik Derneği'nin başkanısınız. Bize birazcık bu arada hazırlıklar tamamlanana kadar kısa bir epigenetik nedir diye bir giriş yaparsanız şöyle 1-2 dakikalık. Ondan sonra da sizi moderatör olarak size bırakalım. İki önemli konuşmacımız burada. Siz bir epigenetik girişi yaparsanız konuda daha güzel anlaşılır diye düşünüyorum. Teşekkür ederim.
Çok teşekkür ederim. Böyle güzel bir organizasyonun bana da vakit ayırdığınız ve yer ayırdığınız için çok teşekkürler. Türkiye'de evet bir ilke imza attık. Nutrigenetik ve Epigenetik Derneği olarak epigenetiği Türkiye'ye tanıtmaya karar verdik. Sizlerin aracılığıyla bunu universal hale getiriyoruz. Her alanda olduğu gibi biliyorsunuz bir genom projemiz vardı. Genom projesi yapıldı. Evet, herkesin farklı özellikleri olduğu, single nucleotide polimorfizme bağlı olarak değişik özellikleri insanlara yatkın olduğunu tespit etmekle beraber proje bittikten sonra yeni bir sürprizle karşılaştık. Bu yeterli gelmediği anlaşıldı. Evet, bir kişinin kansere, kalp hastalığına yatkın olması onun hasta olması anlamına gelmiyordu. Peki neydi bunu etki eden faktör? Epigenetikti. Epigenetik neydi? Bizim DNA'mızı değiştirme şansımız yoktu. Tek nükleotit değişiklikleriyle beraber yatkınlık olsaydı değiştiremezdik. Ama ifadesini değiştirebilirdik. Ne anlama geliyor? Bir muzda da DNA var. Veya işte yiyecekleri bitkinin içinde DNA var. Ama ifade olmadığı zaman bu muz olarak kalıyor. Bizler de bir canlı olarak hayatımızı idame ettiriyoruz. Biz DNA'mızı değiştirmeden ifademizi değiştirerek kalp hastalığı, otoimmün hastalıklar, kanser ve alerjiden kurtulabiliriz. Ve bu nereden geçiyor? Yediklerimiz, içtiklerimiz ve en önemli tedavilerden bir tanesi de sizin uyguladığınız tedavi. Gerçekten çok önemli. Yani şu anda tıbbın değişik kollarındaki arkadaşlarım çalışma içindeler. Integrative medicine diyorlar, farklı bakış açıları sağlıyorlar. Evet, ama bunun gerçek bilimsel tarafı hepimizin yapmak istediği şey epigenetik değişiklikleri, olumsuz davranışları olumlu davranışlara çevirerek insan sağlığına koruyucu hekimlikte hastalıklardan en sağlıklı bir yaşamı uzatarak, yaş alıp yaşlanmayarak ve yaşla gelen hastalıklardan bizlerin korunmasını sağlamaktır. Çok teşekkür ederim. Hocalarımızı da çok bekletmeden arzu ederseniz başlayabiliriz oturuma. İlk konuşmacımız Associate Profesör Doktor Trimulus Kanan. First, please, if you can start your presentation.
Okay. Oh. Thank you, Dr. Stephen, and thank you, Prof. Gulsen, and the organizing committee for inviting me to present my talk on relationship between telomere length and beekeepers. And I'm Kanan from University of Science, Malaysia. So just to give a brief history, like previously, it was believed that beekeepers live longer than other human beings. And to quote some examples, Francois Huber was a Swiss entomologist. He lived for a span of 81 years and was associated with beekeeping activities. And Lorenzo Lorin, the father of American beekeeping, he also lived for a span of 85 years. And Johann, a Polish apiarist, who is credited with the discovery of parthenogenesis in bees, he lived to a span of 95 years. But is this belief only an old wives' tales or vice versa? That's what we are going to see in the current study. But before we actually go into the study proper, I'll just like to highlight some basic description of telomere and its function. So we all know that cell contains nucleus and nucleus contains chromosomes. And this telomere is present at the end of the chromosome, which is a 6 nucleotide repeat sequence, TTAGGG. And the role of this TTAGGG is to provide the stability to the chromosome. So basically, it prevents the end of the chromosome from sticking to each other. So that's the main role. And the telomeres shorten with every cell division. So if you see when the cells are multiplying in the synthetic phase, there will be reduction in the number of base pairs with each cell division. And since telomere length declines with age, it is possible to estimate the age by using telomere restriction fragment length. Therefore, we can estimate the longevity of life biologically by comparing the telomere length between individuals. So this is the telomere, what is highlighted in red color. It can be compared to the aglet of a shoelace, like how the aglet of a shoelace gives protection to the shoelace. Similarly, the telomeres provide the protection to the chromosome. So bearing that in mind, we had two main objectives in this study. One was to determine the TRF length of telomere among beekeepers and non-beekeepers and to statistically evaluate them. And the second main objective was to determine the association between consumption of bee products and telomere length variations. So we had some inclusion criteria and exclusion criteria for the study. First was that healthy beekeepers and non-beekeepers above 30 years of age were recruited. Because if we see the telomere attrition or the decrease in telomere length, it is high during the early childhood. And from the age of 4 until 25, there is a bit of plateau. So only after the age of 25 years, there is a gradual decrease in the telomere length, which is a very consistent way of decrease in telomere length. That's why we chose the age as 30 years and above. And for beekeeping related activities, they should be associated with a minimum period of 5 years of beekeeping, because too short a period, it may not be able to distinguish the base pair difference between both the study groups. And for non-beekeepers, those who do not consume any bee products and are not associated with beekeeping related activities were included. And for the exclusion criteria, both the study groups who had any disease or who are taking any kind of medication were excluded, as we all know that this can influence the telomere length. And prior to collecting blood, we recorded the blood pressure in both the beekeepers and non-beekeepers, which I will explain later. So this is the methodology in brief. We got the ethical approval from the Research Ethics Committee of University of Science Malaysia. And the blood samples were collected from both beekeepers and non-beekeepers. But here, one thing what I want to highlight is we recruited only the males in the study, because in females, the estrogen can influence the telomere length. So in order to avoid the bias, we recruited only the male beekeepers. And then DNA was extracted using a commercial kit. And this was subjected to the telomere length assay. For this, the kit was purchased from Roche, Germany. And the extracted DNA was digested using two restriction enzymes. One was HINFI, isolated from Haemophilus influenza. The other one was from RSAI, that is isolated from Rhodopseudomonas spheroides. And why we chose these two particular restriction enzymes was because they are more affiliated towards the telomeric region of the chromosome. So even they can avoid the subtelomeric region. So that's why we recruited these two restriction enzymes. And after DNA digestion, we ran a gel electrophoresis to separate the DNA fragments which were digested, then followed by southern blot analysis to transfer the DNA fragments to the positively charged nylon membrane. Then this was hybridized using the digoxygenin probe and incubated further with anti-DIG alkaline phosphatase. So when there's a combination of this DIG and anti-DIG, it produces a chemiluminescence, and that is detected on an X-ray film and compared to a nuclear weight standard, which comes with the kit on an ImageJ software. And then later the results were analyzed, interpreted, and statistically evaluated using an independent t-test. So coming to the results, first let us say about the demographic analysis. We collected blood samples from 36 beekeepers and 36 non-beekeepers, but later when we extracted the DNA, we found that 6 were degraded in both the study groups. So the sample size was 30 in both the groups. And the origin distribution of beekeepers, out of the 30 samples, 60% were from Sabah, it's a state in Malaysia, followed by Johor, which was 37%, and then Melaka was 3%. And coming to the race distribution of beekeepers, the majority were Bajau. Bajau is an ethnic group in Philippines, and they have migrated to Sabah and settled there, which was followed by Malays constituting 37%, and then Chinese 3%. So this is the baseline characteristics of beekeepers and non-beekeepers before we actually continued with the study. the age was recorded, and the mean age in case of beekeepers was 44 years, and in case of non-beekeepers, it was 41. But there was no statistically significant difference. And the blood pressure, as I told earlier, it was recorded prior to collecting blood. And why we recorded the blood pressure is because it has been proven in earlier studies that there is a inverse relationship between the telomere length and systolic and diastolic pressure. So that's why we tried to record the blood pressure. And we found that the systolic blood pressure was 137 in case of beekeepers and non-beekeepers with no statistical significance. And for the diastolic, it was 76 and 80 in beekeepers and non-beekeepers respectively, which was also not statistically significant. And this is the gel electrophoresis picture showing the DNA integrity. The B1 to B6 represent the samples from beekeepers, and followed by dissected DNA samples. We had the control, which is from a cancer cell line, comes with the kit. And then B1 to B5 represents the dissected samples of beekeepers, and N1 to N5, that of non-beekeepers. And this is the southern blot analysis results. We compared the results with the DIG molecular weight provided by the kit, which is in kilo base pairs, and control. Then we have the beekeeper samples and the non-beekeepers samples. So coming to the mean difference in the telomere length between the beekeepers and non-beekeepers, it was found that the beekeepers had a longer telomere length, that is 8.13 kilobase pairs, compared to that of non-beekeepers, which was 5.85. And the independent t-test also showed that it was significantly different. That means indicating that the telomere length in beekeepers was longer compared to the non-beekeepers. And we continued the study with multiple regression analysis for few parameters among the beekeepers with regard to age, period of beekeeping, number of years, then the number of bee products consumed, like whether they took royal jelly, honey, bee pollen, beeswax, and so on. We had a questionnaire for that. So we collected the data prior to extracting the blood. Then the period of bee products consumption per year. And we found that there is an association between the telomere length and the period of bee products consumed over the span of years. And for every year of bee product consumption, there was a corresponding increase of 0.258 kilobase pairs in length of telomere. And regarding the frequency of eating bee products per day, that was also associated with telomere length with an increase of 2.66 kilobase pairs. So these two are associated parameters with the telomere length. And beekeepers have longer telomere length compared to non-beekeepers. And it's all established that telomere is an indicator of health status. Longer the telomere, better the health status. And the telomere also reflects the longevity of life. And longer telomere length might indicate longer life, while shorter telomere length is associated with increased risk of mortality. And the main factor which contributes to the shorter telomere length is the oxidative stress. And the reason is being the structure of telomere itself. Telomere sequence contains many guanine prone to be oxidized to 8-oxide-G by reactive oxygen species. And hence, whenever there is increase in 8-oxide-G, there is corresponding decrease in telomere length. And the reactive oxygen species also causes DNA breaks and deteriorates the DNA base pair. And coming to the period and frequency of bee products consumption and its association with telomere length, we know that bee products have very great antioxidant capacity contributed by the phenolic and flavonoids. And the honey also exhibits protective effect in endothelial cells against oxidative stress based on previous studies. And the effect of bee product consumption in the long run has been predicted. However, the main mechanisms on how bee products protect telomere length is still unknown. But still, despite, bee products may protect telomere length against oxidative stress, and increased frequency of eating bee products per day increase protection of telomere length. While little is known about how bee products protect telomere length is associated with nutrition status and healthy lifestyle and diets based on previous studies. And antioxidant properties of vitamin C and E has also been shown to be positively associated with longer telomere length. So with that, we came to two strong conclusions in the study. The mean terminal restriction fragment length was significantly longer in beekeepers compared to non-beekeepers, and the period and frequency of bee products consumption are associated with telomere length. However, this study had a few limitations. One is the small sample size. That was because of the short-term grant. So we could employ only so many subjects. And one more major factor was we didn't evaluate the telomerase activity, the driving force behind the increase in telomere length. So that would have been a very good parameter to be included. And also the study was limited to cross-sectional in nature. That is one time point study. However, the future prospect, it will be better to do on a larger cohort study, that is employing the same subjects, and we take the measurement at different time points, maybe after 1 year, 2 years, 3 years, then that will give a better correlation regarding the telomere length and beekeeping activities. And it will provide a understanding on the roles played by bee products in telomere biology. And the outcome of this study was published in the American Aging Association journal, Age. And here you can see Dr. Stephen Stangachio's name, at last, who gave us the idea of conducting this research when we had an international conference on honey in Malaysia. And these are my selected references. And I would like to thank the short-term grant of my university for the financial support. And with that, I thank you for your attention. Thank you.
Thank you, Dr. Kannan, for your presentation. You have provided some very important information to us. Thank you for your contribution. Very important, I know, is the healthy eating, healthy food, and the healthy information very important in the long life. Thank you so much. Arkadaşlar, gerçekten çok değerli bir konu. Epigenetik, çevresel faktörlerle ve yediklerimiz, içtiklerimizle yaş alabiliriz ama yaşlanmama elimizde. Epigenetik saat olsun, bu artık telomerlerin boy uzunluğu olsun, son zamanlarda oldukça önemli konular ve bu konu hakkında çok çalışmalar var. Hepimizin bir epigenetik saati var ve epigenetik saatimizi geriye alabiliriz, yaş alabiliriz, yaşlanmayabiliriz. Thank you so much, Kannan.
Thank you, Professor.
Now is starting Dr. Jones ve Dr. Katerina. Please, if you can start your presentation.
Kannan, you can stop the presentation. Kannan,
yeah, sure. Yeah,
above, yes. Stop share. Yes.
Okay.
Okay. My friends, I heartily greet you. I welcome you in my native country, Slovakia. According to the old habit, we welcome a rare guest. What are you all? With salt and bread. And I want you welcome with royal jelly protein and royal jelly. Okay, can I?
Yes, and look to the slides. Look to the slides to speak on the same slides.
Okay. The previous.
No, it's a video and it's difficult to go back. Yeah.
It's okay. Gradually is a secretion product of the cephalic glands of nurse bees, and it is unique nutrient developed in nature during evolution of animals. The major part of dry matter of royal jelly are major royal jelly proteins. And from this protein part of royal jelly, the major royal jelly proteins represent 90%. The minority part of royal jelly proteins are antimicrobial peptides and enzymes. Okay, next slide. Can we go on to the next slide?
It will come, the slide comes, yeah, okay. Yes?
Few words to the nomenclature of royal jelly proteins. On beginning our research royal jelly, we suggest terming major royal jelly protein as MRGP. Later, we have found that these proteins are present also in honey, bee pollen, and propolis, respectively.
Joseph, can you hear me? Yeah, I'm speaking from technique. We are also reflecting your record, and
Yes.
I think at the same time you are
Yes. Speaking. Yes. Yes. I understand.
reading your
Yes.
presentation. Yeah.
Okay. Can you
We can stop our screen share and you can continue with your presentation.
Sorry.
One other idea for the technique, and also Professor Simut, we can project your PowerPoint, and then you go slide by slide like normally. Do you have the technician, the PowerPoint of Professor Simut?
Yes.
Yeah. Okay. Then start the PowerPoint, and Professor will say next, next, next. Maybe it's better like this.
I must do it.
Wait a second, maybe the technician can do it. Did you find the PowerPoint? If not, I can make the share screen from me, from my computer.
Anyway.
If you want, Dr. Stefan, if you want, you can start the same.
Yes, yes,
yes.
yes.
I think it's.
She found. Okay.
If you want, stop and continue it changing every.
Yes. You can go to the slide 3 or 4.
Okay.
Okay. Simple. Name of major royal jelly proteins are, next slide, apalbumin. Okay.
Okay.
Next slide. We are focused on the proteomic royal jelly proteins. Major proteins royal jelly is one gene family consisting of 9 genes. Recent study on proteomic complement of royal jelly showed on 2-dimensional gel at least 50 distinct protein spots that were identified by mass spectroscopy as apalbumins. It means that maternal apalbumins form a great number of minority homologs with various post-translation modifications and different physiological properties. Next slide. The apalbumin 1 to apalbumin 4 are dominant proteins, looking here, about 95%. The apalbumin 6 to apalbumin 9 are trace proteins. One of them, apalbumin 9, is also in bee venom. They have no nutrition function and other physiological properties. Next slide. One of these is production of cytokines. For example, apalbumin 1 in monomeric form has the same immunostimulation activity like model substance lipoposaccharide. Apisimin is peptide and lowest activity, apalbumin 2, and etc. Next slide. These examples show how different physiological activity have one molecule of proteins. Apalbumin 2 is maternal protein of royal jelly. Apalbumin 2A is isoform of apalbumin 2. And this apalbumin 2A has such antimicrobial activity like antibiotic tetracycline on the growth of Penibacillus larvae. It means the spectrum of physiological activity of proteins of royal jelly is very broad. Next slide. It is overview on the multifunctionality of apalbumin, nutrition, antibiotic activity, immunostimulation, anti-hypersensitive activity, proliferation, suppression, and anti-cancer activity. Next slide. Royal jelly is complex biological matrix which is structure is formed by individual honeybee as small particles and secreted to the larvae. Next slide. The electron scan microscopic examination of royal jelly layer showed relatively large spherical particles which are connected to one another by system of filamentous channels. The crucial factor which has influence on the structure of royal jelly is self-assembled property of apalbumin 1. The royal jelly is roughly defined as emulsion, but in principle, in native form, it is like small egg, which is produced by honeybee to the larvae. Next slide. I have isolated and prepared the apalbumin 1 in oligomeric form, 102 kilodaltons by ultracentrifugation. And the self-assembled properties and structure is roughly similar, the apalbumin 1, to them which we observed in the native royal jelly. Next slide. This oligomeric structure of apalbumin 1 and self-assembly properties use the honeybee in processing nectar to honey and immobilization of pollen dust to pellet. The membrane of apalbumin 1 uses the honey and the evaporation of water from nectar. Important is that the royal jelly protein, apalbumin 1, is regular component of honey. Second, this web structure of apalbumin 1 use the honey bee to immobilization the pollen dust to pollen pair. It means the pollen contain apalbumin 1. Next slide. It means the apalbumin is a regular component in honeybee and honeybee pollen. We have developed quantitative method, ELISA, for detection of apalbumin 1 in honey, bee pollen, in other bee products. And all honey sample and the honey have immunostimulation activity. Important is the honey from saccharose syrup has minimal effect on stimulation of cytokine because low amount of apalbumin 1. Depronoside honeys didn't stimulate production of cytokine. Important is the apalbumin 1 is honey-specific protein, physiologically active, and therefore a suitable marker for determination of authenticity of honey. Next slide. We are on the way from molecular apidology to apitherapy. The sequences of honeybee genome opens new territory of honeybee epigenetic in apitherapy. For research on physiologically active proteins and antimicrobial peptides, an application as immunomodulator, suppressor of allergic reaction, anti-hypersensitive agent, alternative classic antibiotics, and epigenetic factor. Next slide. Honeybee genome is surprisingly relatively poor on gene. For example, honeybee contains only half amount of genes which coding protein to human genome. The honeybee are very poor on antimicrobial genes which synthesize antimicrobial peptides. To other insects, only 1/3, 1/4. Honeybee have developed a specific defensive system based on multifunctional proteins and peptides of royal jelly. Albumins act as epigenetic factors. Next slide. What is epigenetics? Epigenetics is the study of how your behavior, aging, for example, an environment, nutrition, drugs, radioactivity, toxic substances, can cause changes that affect the way your genes work. Genes can be turned on or off, that not change the DNA sequence by several types of chemical modification. Most complex diseases such as cancer, mental disorders, central type of metabolic anomalies, and many others are not caused by change on the genetic code, but have an epigenetic nature. Next slide. The most frequent epigenetic DNA modification is cytosine methylation. It means the cytosine, which is methylized in the DNA change, turn the gene on or off. Next slide. Epigenetic system is very complicated. It is not only the methylation of cytosine, but they also modified in the epigenetic processes the histone tile of the chromosome. DNA methylation and histone modification are the most target changes, targets during epigenetic modification. The big genome represent a rarity among the genomes. The queen bee worker are formed from exactly the same DNA sequence by action of external epigenetic factor in royal jelly. The queen and worker, previous. They differ in appearance, perform completely different jobs, with the queen living 10 times longer than the worker bees. The phenomenon of lifespan of honeybee queen made a basis for opinion that the royal jelly might be means for long-living human beings. The genome of honeybee responds very flexibly to the nutritional signal during larval development of bee. And this process can help us to explain the cause of epigenetic changes during human life, which we refer to as epigenetic aging. Next slide. Only shortly. Until now, we know the nutritional signal, epigenetic factors, or in royal jelly, causes different epigenetic methylation of DNA in more than 500 genes during larval development. And second, there are also different modification of histone protein between the worker bee and queen. The Honeybee offer is the applicable model for studying the role of epigenetic factor in development and behavior. Also, a bee cannot be used as model for specific human diseases, but it is offered as research model for understanding the general mechanism of epigenetic processes. Next slide. Honeybee's model in epigenetics in two roles. In the first, it presents itself as model for understanding the general law of epigenetic, because in medicinal research is the non-satisfactory experimental model in animal models. In the second role, it acts as source of epigenetic substances that have a beneficial effect on human health and longevity. We can consider these substances as potential drugs, and I will tentatively call this drug as epigenomicum. Next slide. The news on the apalbumin's effects on the longevity, spatial memory in hypertension. Female Drosophila melanogaster whose food enriched with 2.4% apalbumin living up to 20% longer than those whose diet didn't contain apalbumin. The life of males extended the effect apalbumin by 12%. Extension of Drosophila melanogaster life was accompanied by increased fertility. Apalbumin have effect on spatial memory. Aged male rats fed apalbumin showed improved spatial memory roughly 50% when compared the control male aged rat with distilled water. And finally, apalbumin 1 decreased hypertension via action on most vascular small muscle cells. The apalbumin 1 gene was using by method of gene therapy was inserted in most vascular smooth cells. It is first time which was using gene therapy for confirming one Honeybee protein activity. The antihypertensive activity apalbumin 1 by this expression significantly reduce contraction and proliferation of these cells. Interesting is that apalbumin 1 gene, after expression, it means the molecule apalbumin created by self animals, like in the smooth muscle cells, influence activity of 400 other mouse muscle cells. It means it is new approach to verification of honeybee product by using gene therapy. Next slide. Conclusion. Our approach to honeybees is being rewriting. It changes from honey producing animal to a scientific model that helps man find the fullness of life in a new stage of human development at higher spiritual level. It gives us as epigenetic drugs as epigenomicum derived from royal jelly with slowing or even stopping aging and eliminating diseases in old age. It was presented in previous presentation. And question, my question for apitherapy, for apitherapists, are we ill because we are age? Thank you for your attention.
Thank you. Thank you so much, Dr. Simoti. Your presentation, you have provided some very important information to us. Very different, very nice. Now, Sevgi question. Sevgi Hanım'ın verdiği bir soru vardı, ona yanıt verebilirim. Sevgi Hanım'ın verdiği bir soru. Acaba nasıl bir değişiklik yaratıyor? Epigenetik olarak da ifade etmişlerdi. Şöyle ki, çok güzel Dr. Simoti ifade etti ama epigenetik çok bilinen bir konu değil. DNA'yı değiştirmiyorsunuz. DNA üzerinde adenin timini değiştirmiyorsunuz ama kraliçe arı olaması için, kraliçe olan kişilerde DNA'da daha enerjik, daha genç, daha canlı olması gereken genleri daha aktif çalıştırabiliyorsunuz. DNA üzerindeki kimyasal olayları, DNA modifikasyon ve DNA metilasyon, histon modifikasyon ve non-kodik RNA denen epigenetik mekanizmalarla o DNA üzerindeki çalışması gereken, enerji sağlayan, daha genç olmamızı sağlayan kısımlardaki aktiviteyi daha da arttırabiliyorsunuz. Yanıt verebildiğimi bilmiyorum Sevgi Hanım sorunuza. Thank you so much, Dr. Simoti, Dr. Kanas. Now, I look at question, another question.
Yeah.
I don't see this another question now.
Maybe one remarks to the previous lecture, Professor Kaman.
Yeah.
The
Can I answer?
apalbumin 1 and royal jelly proteins, mainly apalbumin 1, has antioxidant activity, very high. And the antioxidant activity is the main reason why the queen life is so high. And consumption of honeybee product, it is no consumption of royal jelly, but the apalbumin 1, the royal jelly proteins are present also in honey and bee pollen. It means the antioxidant activity are present in all honeybee product, and it correspondent with your finding. And that was my comment.
Dr. Stephen, close your, I don't hear you.
No, I just wanted to stimulate Professor Kanan to answer because he wanted to give a comment about correlation with royal jelly.
Not royal jelly, because I found one question in the Q&A box, because one of the panelists or participant wanted to know whether the non-beekeepers were consuming bee products or not. So I just wanted to clarify regarding that, because here the beekeepers were selected who were engaged with beekeeping related activities, and surprisingly, some of the beekeepers, even though they are engaged with beekeeping, they do not consume any bee products. That is number one. And number two, the non-beekeepers, they were not associated with any beekeeping related activities, nor were they consuming any type of bee products. So it is totally nil. So that is how we compare. Thank you.
I appreciated this experiment, and it is based for new orientation of apitherapy using discovery in basic biology and molecular genetics. We are most looking at some signals. And for example, the longevity of queen is not derived. They have the potential for elimination of free radicals genes, which are which have activity for elimination of free radicals. The genes in queen are still working on very low level. For example, to worker bees. Why? Because the royal jelly proteins play the crucial role in antioxidant activity.
Yes.
Okay. Thank you so much for contribution. Thank you so much.
The gene is not so strong during the life, but the royal jelly proteins are very strong in apitherapy.
Yes.
Okay. Thank you so much. I'm sorry speaking Turkish. Çok önemli bir ayrıntıyı hazır fırsat bulmuşken herkese buradan anlatmak istiyorum. Nütrigenetik ve nütrigenomik çok karışan iki ifade. Şimdi arkadaşlarımız soru sormuş, herhalde tam ifade edemedik. Soruyu belki tekrar alabiliriz Dr. Stephen. Diğer arkadaşlarımız nütrigenetik ve nütrigenomik. Nütrigenetik, genetiktir. Yani genetiği çok değiştiremezsiniz. Nütrigenomik esasında içindedir epigenetik. Yani yedikleriniz içtikleriniz ise epigenetik olarak DNA ifadeni değiştirebilirsiniz. Yani epigenetik, yediklerimiz, içtiklerimiz epigenetik ayrı bir şeydir, nütrigenomik. Nütrigenetik ise DNA'mıza uygun. Yani bir kişinin enziminde bir defekt varsa o kişinin ona uygun beslenmesi lazım. Single nükleot polimorfizmde. Bu nütrigenetiktir. Ama epigenetik nütrigenomiktir. Yani kimyasal dışarıdan mikrobiyota da bunlar içindedir. Yediklerimiz, içtiklerimiz ile DNA üzerindeki ekspresyonumuzu değiştirip uzun yaşam sağlayabiliriz. Burada da tabii Rajay'ın çok önemi var. Biz de epigenetik ve nütrigenetik epigenetik derslerimizde, eğitimlerimizde bunu vurguluyoruz. Katkınız için çok teşekkür ederiz. Thank you so much, Dr. Smith ve Dr. Kanas.